Thank you everyone for attending today's webinar, where we will be discussing a scalable solution for the creation of lipid nanoparticles for RNA delivery. My name is Chris Jaquin, and I am an applications engineer at Microfluidics. I will be serving as today's host and will also be briefly presenting. Our featured presenter today is Dr. Derek Carter, Chief Scientific Officer at HDT Biocorp. To begin, we will briefly go over the agenda for today. I will begin by giving an overview on Microfluidics, discussing who we are as a company, giving a brief applications overview, touching on how Microfluidizer technology works, and also briefly discussing our Microfluidizer product line. At this point, I will hand over the presentation to Dr. Derek Carter, where he will be discussing the history of RNA vaccines, major delivery systems currently in use for RNA, how self-amplifying RNAs work, his line carrier, cost considerations for scale-up, using microfluidics devices to scale from bench to commercial production, and future directions of this technology. At the end of the presentation, we will have a question and answer session, so feel free to ask any questions you might have in the chat feature throughout the presentation, and we will get to as many of those questions at the end of the presentation that we can. So to begin, I will be discussing who Microfluidics is as a company. So Microfluidics was founded in 1982, and we have been manufacturing high shear fluid processors utilizing our proprietary fixed geometry interaction chamber technology ever since. We are headquartered in Westwood, Massachusetts, just outside of Boston, and we have localized support in 47 countries. To date, we have sold over 4,000 processors to over 2,000 companies. Around 10 years ago, we were acquired by the IDEX Corporation, and have since been grouped with our sister companies Quadro, Fitzpatrick, Matcon, and Sterados to form the Materials Processing Technologies, or MPT platform. Our processors are utilized in a variety of industries for a variety of applications, including for both R&D and manufacturing of products such as APIs, vaccines, inks, coatings, nutraceuticals, cosmetics, and much more. Over our 40 years of existence, we have amassed vast experience in both machine design and applications. Speaking of applications, I've listed a handful of the more common ones that we see on this slide here. Today, we will obviously be discussing lipid nanoparticles, and similarly, our technology can also be used in the manufacture of both nanoemulsions and liposomes. I have also listed some of the more common applications that we see on this slide, including polymer nanoparticles, cell disruption, polysaccharide molecular weight reduction, deagglomeration, and particle size reduction. It's important to note that while these are some of the more common applications that we deal with a plethora of other applications as well. So how does microfluidizer processor technology work? To begin, you will introduce your product into the inlet reservoir, where it'll be then introduced into a constant pressure pumping system, which forces your material through the fixed geometry interaction chamber at pressures of up to 30,000 psi or around 2,000 bar. Within this fixed geometry interaction chamber, your material is subjected to constant high shear rates and high impact forces. As it is a fixed geometry interaction chamber, all material receives the exact same treatment, which allows us to achieve consistent, reliable results. After your material is passed through the fixed geometry interaction chamber, it is then effectively temperature controlled and then collected. We offer two different types of interaction chamber designs, as can be shown on this top graph. One of the types is a Y-type interaction chamber. The Y-type interaction chamber works by having your product enter through the high pressure inlet before separating into two streams. The two streams then pass through two separate microchannels where they are subjected to high shear forces, before then impinging upon each other in the high impact zone. Within this high impact zone, the two streams lead to intensive micro mixing, and this is ideal for liquid-liquid dispersion. So you can think of lipid nanoparticles, nanoemulsions, and liposomes. The other type of interaction chamber that we offer is a Z-type interaction chamber, and it differs, as you can see, in that there is only one microchannel, and the high impact zone is up against this wall rather than the two jet streams impinging upon each other. The Z-style interaction chamber is most suitable for any systems that contain solids, so you can think suspensions, dispersions, or cell disruption. But one of the major benefits of microfluidizer processing technology is that it is completely scalable. In order to do so, what we do is we simply place the single microchannel that is utilized on the lab scale in parallel on the production scale. By doing so, we ensure that as the individual microchannels are all the exact same, that so long as your formulation and processing conditions are the same, they will be subjected to the identical high shear and high impact forces within each of the individual microchannels, thus allowing us to guarantee linear scale-up results. On this slide, I have listed our product portfolio, and as you can see, we offer a wide range of products from our lab to pilot to production scale units. Similarly, we are able to hand a wide range of batch sizes, starting as little as 1 to 2 milliliters on our smallest lab scale unit, all the way up to several thousands of liters on our production scale units. It's also important to note that we offer pharma grade and cGMP compliant models at every scale, from the lab to the pilot to the production scale. And thus, our processors can be used starting at phase 1 clinical trials all the way up to full-scale drug manufacturing. At this point, I will be handing over the presentation to Dr. Derek Carter. But if you have any additional questions about microfluidics, who we are as a company, our applications, our technology, or our product portfolio, feel free to ask in the chat feature, and we will try to get to it at the end of the presentation. Thank you.
All right, thanks a lot, Chris. Let me know if is this showing up okay?
Yep, we can see it.
Okay, great. So thanks for inviting me to this talk. I'm excited to talk about some of the things that we've been doing here. I thought, because people might not be completely familiar with all the mechanisms and where we're coming from, I thought to give a brief overview of the RNA vaccines, then talk a little bit about these platforms that are out there, have some hypotheses about how these different platforms might work, and you know, have some opinions about some of the debates that are going on in terms of modified RNAs. And hopefully by the end of the talk, you'll be able to kind of form your own opinion about what's going on with these vaccines, how scalable they are, and if you're trying to develop your own, maybe this will give you a lesson of how to do this. Finally, I'm going to end up with the issues of scaling and how we've used, with our partners in India, we've used microfluidics technology to hopefully achieve a process that's commercially scalable and could make, you know, up to hundreds of millions of doses. But I'll start with where we've come from and give a brief history of RNA vaccines. Now, this is a simplified picture of what was going on. There actually have been a number of actors in this field who have developed or contributed to the technology. So in 1961 was the first report of mRNA as an agent that could be the carrier between DNA and protein. So when people realized that, they started working on, well, how can we use mRNAs to make proteins? And in 1978 was the first reported version of an RNA in a liposome. Things started accelerating a bit after that, where people were then able to deliver RNA to human cells in this liposome, and then prove that these cells were then sort of reprogrammed to make whatever the RNA was encoding. A couple of years later, in 1993, was the first report of an mRNA vaccine. It was in mice, and it was the application was for a flu vaccine. Then it took a while between 1993 and 2013, and this is kind of the time when most of these companies were formed around RNA delivery. And then in 2013 was the first report of an mRNA vaccine trial for rabies. Now, due to the coronavirus, we made a big leap between 2013 and 2020. Normally, this would take a lot longer between your first proof of concept and having an approved drug. But in 2020, as you very well know, was the first emergency use approval of an mRNA vaccine, and followed up by now complete approval by the FDA of an RNA-based vaccine. And this is drawn from a very nice little report, if you look in Nature for last week, the tangled history of mRNA vaccines. So if you think about RNA delivery, and you want to use some kind of carrier to protect the RNA, since as you well know, if you've worked in the lab with RNA is quite labile. There are a couple of different solutions you could use. These are probably just the major ones that we're envisioning right here. If you look, there's a bunch of different other particles and different forms of RNA delivery, but I've been focusing kind of on the larger ones that are most common. So in almost everything you see, you're going to encounter a lipid nanoparticle. And what that is a particle where there is a composition of lipids and detergents that surrounds the RNA and thereby protects it from the action of RNases and from hydrolysis. That, of course, is pretty convenient because you have only a single vial. However, it's pretty complex to manufacture because you have to combine the RNA and the lipid, and you have to manufacture those in a single process to get to a point where the RNA is encased in the lipid. And so for every time you want to make a vaccine product, you're going to have to encapsulate it on its own. Now, a possible solution to this was developed at Novartis, which is a cationic nanoemulsion. And these were a step further from their adjuvant formulation of MF59, which was an emulsion. And here the RNA is thought to bind on the outside, on the exterior of the cationic surface of this emulsion. This is a 2-vial presentation because the emulsion is stored at usually about 2 to 8 degrees, and the RNA is stored at -80. You mix the two just prior to injecting it. The RNA binds to the surface of the nanoemulsion and then is ready to be delivered. This, of course, allows stockpiling of formulation next to the RNA, and you can do bedside mixing, which is pretty convenient if you're trying to do different targets. Now, we have evolved this another step further in that we've taken the cationic nanoemulsion. We've modified the surface chemistry quite a bit, and we've added a solid core using inorganic oxides. And I'm going to comment more about why this step might be really important to the enhanced stability and bioavailability of our particle compared to some of the particles that are out there. Again, so you have a number of different formulations to choose, but if you're looking at the leads, they all are basically forms of different LNPs. So both Pfizer, Moderna, CureVac, and Arcturus, which are maybe the bigger players in this field, all use lipid nanoparticles in their formulation. They each have a bit of a secret sauce. BioNTech has been using modified RNAs, and I'm going to talk more about what this modification is. Moderna, that's basically how their name evolved, modified RNA. It's basically a compression of those two words together. CureVac has been proposing that due to their unique formulation that they can use unmodified RNAs. And finally, Arcturus, with their lunar platform, have been focusing on self-amplifying RNAs. So let's think a bit about the biology and maybe come to a conclusion about how these different platforms can work and whether or not you need a modified RNA. Now, LNP delivery occurs through an injection in the muscle. That injection causes a local inflammation, and if you've had any of these major ones, you probably felt the local inflammation at the first shot as kind of a point, like somebody had punched you maybe in a small point in the arm. And then at the second shot, you might have felt a little bit larger symptoms of transient local inflammation at the injection site. So this inflammation causes antigen-presenting cells to come. They pick up the vaccine product and they drain to the draining lymph node. And in that draining lymph node is where T cell priming and B cell maturation occur, and as well as presentation of the antigen. So, of course, the lymph node is a site where there's a lot of immune activity and immune cells. So if you're stimulating innate responses, that might be a place where you could get a lot of inflammation if you don't have a modified RNA. So if you're looking at somebody like CureVac, you can see that, you know, in the press, they had relatively underwhelming results with the vaccine efficacy about 48% compared to Pfizer and Moderna's 90 something, 95, 94, depending on who you think. But you also look at the top dose that they chose. So they chose a 12 microgram dose. And really what I think is happening is they are between a rock and a hard place. And the reason for that is that they are using an unmodified RNA and not using these synthetic nucleotides in the formation of their RNA. Now, normally our cells have an antiviral program that when a RNA enters the cell, it can get sensed either in the endosome through the various toll-like receptors, TLR3, TLR7, and TLR8, or if it escapes the endosome and gets into the cytosol, it can be detected by the RIG-I pathway or MDA5. All of these pathways ultimately result in triggering NF-kappa B, and this program is an interferon-based program that then suppresses translation. So you get stalled translation, you get inhibition of replication, and you get degradation of the RNA, as well as local inflammation. So triggering these innate sensors is not a good thing for an mRNA vaccine. And this just shows you that these interferon-inducible genes, if you don't cap the RNA properly, if you don't have if you have unmethylated RNA, or if you don't have a 5' triphosphate, in each case, these interferon-inducible genes will start blocking translation. Not only will they do that, they'll also stop translation by blocking initiation of the translational machinery here. So this is why, of course, you might think of modifying your RNA, and here's one example of what these methylated uridines will do. In that, if you look at the TLR7 structure, TLR7 binds uridine in its recognition pocket. And when it's bound nicely, as shown in the crystal structure here, it can cause signaling of TLR7 and release of these interferon genes. But if you have this methylated RNA, you get a steric hindrance. So this uridine is not able to bind tightly to TLR7, and thus suppresses the signal of TLR7 and reduces the amount of inflammatory and activation of interferons. So this is where the whole idea of these nucleoside modifications come from with these pseudouridines and the whole discussion of whether or not the Nobel Prize should be given for that. But, you know, I think there's many ways to skin this cat. So, you know, capping is probably going to be very important, but I'm not sure that you necessarily need the nucleoside modifications. Now, that is kind of what CureVac said too. They said, well, you know, we might not need them if we formulate our thing properly. But I think because of the way their platform works, that they're stuck between two things. They could either increase the dose to increase the response. However, when they do that, the increased dose leads to more activation of innate pathways and shuts down the translation. So at some point, you can you can't increase anymore, and if you decrease, you might not get the appropriate immune response. Okay. So if you, though, change your delivery platform and use something like a cationic nanoemulsion, the cationic nanoemulsions don't seem to drain as rapidly to the draining lymph node. They actually transfect these muscle cells, and this is from a paper from Novartis's group with Derek O'Hagan, cationic nanoemulsion for the delivery of next generation RNA vaccines. And what you can see here is a muscle section after giving a cationic nanoemulsion RNA that encoded for a beta-gal reporter so that they could get this blue color in the section and show that the muscle is actually producing the protein that they put in their vaccine construct. Now that's a good start, but I think you can go even further. And what you could do is use a self-replicating platform, and that's the path that we have chosen. And what we do is we have a proprietary delivery system We bind the RNA to the surface of that delivery system, kind of as I talked about, and then when it gets injected, it binds to the surface of the cells. After a while, we now know it's probably between 1 and 8 hours. These nanoparticles are taken up within the cell, and they dump their payload into the cell, and the RNA is released there. So after release of this replicon RNA, there's a primary program that gets triggered that produces viral polymerases. These polymerases then cause amplification of the delivered RNA. And this amplification then yields more and more of we call the subgenomic RNAs, which are the portion of the RNA that you're interested in. So this is where the gene of interest is cloned into. And this gene is then reproduced a number of times. So if you had an mRNA, you get a 1-to-1. For each mRNA you deliver, you have one message inside the cell. For a self-amplifying RNA, for each message you deliver, we think you get 10 to 50 copies within the cell of your target RNA. And so that target RNA is then read by the translational machinery, and then depending on the types of signals you put on it, in this case we have a membrane anchoring signal, the resulting protein gets transported to the cell membrane and then gets presented to immune cells at the surface of the cell. And this would then trigger the B cell and T cell maturation programs that I talked about. So that might actually be a way out, because you can now use a much lower dose. You can use about 10 to 50-fold lower amounts of RNAs, even if you're using an unmodified RNA in this case, because you get that internal amplification by the body itself. And then on top of it, because rather than draining to the lymph node at a site that is highly immunologically active, you localize to the muscle, and so you reduce the inflammatory responses. So this really might be a way out of this conundrum of increased dose and activation of innate pathways. And sure enough, if you look at what we've seen, here's an example of one of our papers in non-human primates, where with just 2 doses of low amounts of RNA, so 50 or 25 or 50 micrograms in the primate, you can see that you get a very nice development of an antibody response, and that response is boostable about 6 months later. If you use a high dose of RNA, you don't even need the boost. So after just a single injection, you get 6 months worth at least of production of antibodies. So we think that there's a lot to be said for this replicating RNA platform, and non-human primates are a little bit more resistant than we think human primates are. So in our clinical trials, we've gone down in dose. So now I'm going to talk about the delivery platform that we've developed, and I mentioned a little bit, it looks somewhat like a cationic nanoemulsion in that it has ionic surfactants at the interface between the aqueous phase and the oil phase. And those cationic surfactants are thought to be the portion where the RNA binds. We also have an oil core that's immune stimulating, and we have these solid particles inside the lion emulsion. And these are composed of superparamagnetic iron oxide nanoparticles. The original thought behind this was that we would get MRI signals and we were going to go into cancer applications, and so we would be able to follow our vaccine using standard imaging technology. However, now that we've looked at it in a couple of animal models, we have this surprising finding that we actually get really, really good immune responses. And so here in the bottom is an image of a lion particle. You can see that oil core and the slightly lighter TEM signal, and then these very electron dense portions are these superparamagnetic iron oxide particles. And then, as I told you, these particles then localize to the surface of the cell, and here's an image, shortly after injection. I think it's about an hour after injection. You can see these particles at the surface of the cell, and they trigger endocytosis, and they get brought into the cell. This is a much different amplification here, and then you can see what we think is kind of an endosomal compartment where the cell has engulfed these nanoparticles, and these are on their way to being dumped into the cytoplasm where they'll be read. So the manufacturing process to produce these nanoparticles is fairly straightforward and scalable. So you take these iron oxide nanoparticles, they're usually delivered with an oil. So we have to wash that oil off. We use a solvent to get rid of the oil. Then we expose them to an oil phase made of squalene, a cationic lipid, and an anionic surfactant. And then we blend them with a high shear blender. And that just causes them to form these larger bubbles and causes good mixture. And then we feed them through one of the microfluidizer devices in order to get to the final particle size of between 50 and 70 nanometers. And once they're at this particle size, they're quite stable, and this size is quite good for antigen uptake by antigen presenting cells. So we think this is an optimal form of this delivery system. And when you do this, and when you add RNA, you can see that the particles grow slightly. So they grow from that 50 to maybe 80 to 100 nanometers. And when they do so, though, they start protecting the RNA from destruction. So the RNA becomes more thermostable. You can leave it at 25 degrees for at least a week. And the particle size is quite steady, even at higher temperatures, for 7 days. So, this would be good if you wanted to make vaccine and then maybe ship it to clinical sites. You could even ship it at room temperature, essentially, and as long as it's used within a certain period of time, the RNA could be stored in very, very amenable conditions for global deployment. But what you can see here also is that once you inject it into the body, the RNases are going to attack it, and then if you have naked RNA, it's almost gone instantly. However, with our particle, even in the presence of added external RNase, the RNA is completely protected. So why do we think that these solid cores are important? This goes back to a little bit of thermodynamics, and there is a little bit of hand waving in this argument, but we do have to try to figure out why this might be superior. So if you look at the free energy of a system, the free energy is composed of the enthalpy or binding energy of that system and the entropy component. And this entropic component is actually highly temperature dependent. And so if you have a system that is where most of its energy is derived from entropy, you have a much more temperature dependent system. And so you might be much more at the mercy of a cold chain. Now, what happens is these iron oxide nanoparticles are positively charged, and we think they can localize the RNA to the surface. And what you exchange there, instead of having this big rope that's bouncing on a somewhat deflated beach ball, which is very high in entropy as a CNE would be, you start tethering the rope to a solid particle, and that tether causes the formation of a binding energy, so an enthalpic component rather than an entropic component. And so essentially what you're doing is you're shifting the total free energy of the system away from a free energy that's dictated by entropy, and you're shifting it towards a more enthalpic system. And by doing so, you're increasing the thermal stability of the entire system. So I hope I gave somewhat of a background of both what our platform is and what the various RNA vaccines are doing. So now I'm going to shift to our scaling. We had some, pretty good preclinical data, so we wanted to start moving this in collaboration with our partner in India into a scalable solution that could be deployed, not just in the US, but in countries that are still clamoring for vaccines. India, for example, has 1.2 billion people. So you can imagine making 2.4 billion doses is going to be a problem for any single pharmaceutical company. But there are some things that you encounter that are quite staggering when you think about the raw material sourcing for scales at that scale. So here's one of my favorite examples, because I used to work with S-adenosylmethionine as an undergrad, and we used to buy this in small quantities, like 250 microliter quantities that cost about $200. It's a very labile compound, so you have to be careful when dealing with this. So this is at that lab scale. At commercial scale, this same compound, we've predicted now we're going to require about 4,400 liters of it. So, that's a staggering quantity. So 30 liters of nucleotides, 550 liters of GTP, and 4,000 liters of S-adenosylmethionine are going to be required to get us to our commercial scale. Now, if that pricing had scaled the same way, we would need about $3.6 billion just to buy one of the reagents to make the RNA. And you can imagine that this problem is amplified across all the different components that we need for in order to produce the vaccine at this very, very large scale. So we're addressing this with a lot of vendors have been very good to talk to us about scaling to hundreds of kilogram scales. And you can certainly find vendors like Thermo or so who can provide these molecules at these scales. Then once you have all the molecules, you also have to manufacture at the same scale. And this is really a tale of the various microfluidics devices and how nice it is to work with a system that goes from the benchtop all the way through commercialization with very little change in terms of its chambers or devices. So we started with the M110P, which is a benchtop microfluidizer system, and it can run at about 100 mils a minute. Our batch sizes were only about 100 mils, so we could process this in a very suitable time scale. Next, we worked with our partners to do tech transfer, and they transferred from the 110P to the 110EH, which works at about a 400 mil per minute size, and they were working at a 500 mil batch size. Now, one thing I need to say is that when I call this the batch size, this is a concentrate, a 10x concentrate that we produce. So we produce the emulsion at a somewhat more concentrated form. And the reason for this is again, a thermodynamic reason, is as you increase the amount of oil and detergent, the percentage, these particles actually coalesce faster. It's sort of counterintuitive, but it's actually easier to make emulsions at a higher concentration. And so what you can do then is you can make this, for example, here at a 10x scale, and then dilute it out in order to make many more doses than you're actually passing through the microfluidizer. So this 500 mil batch size actually translates to a 5 liter size. Now our partners then were able to scale it to the M815, and that works at 1 liter a minute. And now the batch sizes are growing to 10 liters, which after dilution works out to about 100 liters of vaccine product. And then ultimately where we're going to end up is with the M700, where we're going to work at a 4 liter per minute scale. Oops, I was going to fix that. That's supposed to say 100 liter batch size. And now you're really talking batch sizes where you can produce enough in order to cover the pharmaceutical demand for your product. So as I'm speaking right now, we've gone through the scale up all the way through the 815. What's currently ongoing is the scale up to the M700. And to give you a perspective of where we were at the benchtop scale, we were at 1,000 to 2,000 vials. Once we did the tech transfer, we're between 5,000 and 7,000 vials. Now this 815 can do 1 million doses. So you're already talking, early commercial scale or enough for registration trials. And then once we get into the 700, we're talking hundreds of millions of doses. So basically covering the continent with vaccine in a year. Now, unfortunately, I can't report our clinical results. We're still on a hold. We have received the clinical results, but I can say that even at the lowest dose that BioNTech is envisioning, and actually below that, we're seeing very, very good neutralizing responses in humans. as you could expect with a vaccine that's based on a replicon. And you know, the protective efficacy, this is interesting from a paper here on the right, where if you compare the neutralization level of your vaccine in the lab to the neutralization level of a convalescent patient, you can almost predict the field efficacy of your vaccine. And so if it's identical, this is where you get to about where the chimp adeno or so vaccines are. You get about to 60 to 70% efficacy. And as you go into multipliers of this, you get up to these 95%. And we think we are, based on our lab data, we're probably up there where we really want to be. So now we're going to go back to our roots, and here's an example of immune therapy. This is a patient who has had lesions in their liver, and she was basically cured using a personalized cancer vaccine. And you know, she was given just a month to live. She got these vaccine shots, and 14 months later, she's completely clear of cancer. So this is where we would like to go, is where we take a patient, sequence their genetic information, and then inject them with the vaccine. And because of the way the system works, we could have the carrier manufactured prior to the RNA. We would just order the RNA. It would then be delivered to us, and within a week, the patient could be injected with sequences that were identified from their own tumor. So this is where we're heading. So thank you very much, and I'll hand it back to Chris.
Yeah, thank you so much to Dr. Carter for the incredibly interesting presentation. Before we open up the floor to questions, we just wanted to, again, show our contact information and indicate that if you have any additional questions outside of this presentation, that you can feel free to contact either Dr. Carter or myself. If you're interested in learning more about the LION platform, obviously feel free to reach out to Dr. Carter. And if you're interested in learning more about microfluidizer processor technology, feel free to reach out to myself. At this point, we will begin the question and answer portion, and I see there are already some questions in the chat. So I think we'll just start with the first one. So the first one is for Dr. Carter, and the question is, do the iron oxide particles act as adjuvants within your system?
Oh, that's a great question. And unfortunately, the response is less interesting. It's I don't know. As you probably know, because you're asking the question, is iron is used in Fenton-Weiss reactions inside antigen-presenting cells in order to suppress intercellular parasites, and thereby can trigger adjuvant pathways. But we haven't proven yet that these particles do this. We do hope it does, because it really contributes to the vaccine component of it, but we're still investigating.
Thank you. And then the next question is siRNA inside or outside of the LION particles?
The way I think of it, and based on some really ugly micrographs, it looks more like a semi-submerged submarine. So if you think, if you're standing on these particles and swimming in the dough tap that's on the surface, you would see this huge submarine that's partially encased in the dough tap and possibly interacting with the iron oxide nanoparticles, but other portion is exposed to the surface. And so this is why we think that the RNases can't chew it up, because they can't enter this hyper oil core.
Okay, thank you. The next question is, how large is the RNA? Are they sensitive to shear, and will microfluidic machines fragment the RNA?
No, that's a great question. And you know, I suspect that's why some companies who use LNPs don't go into self-amplifying RNAs. You know, these RNAs are, of course, thousands of kilobases. And if you would microfluidize them, you could shear them quite a bit. However, as I mentioned, we have a two component system. So we only microfluidize the core particle, and we don't include the RNA in that microfluidization. And so we add the RNA afterwards to it, so it doesn't experience these shear forces. So we're not constrained by the size of the self-amplifying RNA. And as a matter of fact, you can clone very, very large things into these self-amplifying RNAs.
Okay, And then the next question is, you mentioned that your clinical trial is still on hold. Was this a hold imposed by a regulatory body? Then they're wondering what might be the reason for the hold.
Sorry, it's not on hold. It's actually in phase 2 trials. I just can't discuss the data yet. The partner wants to, they want to make a big splash with their data, so they've asked me not to reveal what they found. It tells you though something that we did get the green light from the Indian authorities to go into phase 2 and potentially phase 3 registration trials. So
great, that's very exciting. The next question I can handle, it's asking which type of interaction chamber, the Z type or the Y type, is better for LNP production. On the microfluidic system, really any sort of liquid-liquid dispersion, such as the lipid nanoparticle, nanoemulsion, or liposome. For all of these sorts of products, we would recommend utilizing the Y-style interaction chamber. The next question that we have listed is, what is the maximum RNA size that the Lion particle can hold?
We haven't encountered a limit yet. We've cloned different viral genes, cancer antigens, very long cancer antigens, as a matter of fact, in there. Things that have 12 transmembrane domains, and we still haven't encountered an upper limit. What was I going to say? Oh, the thing to mention about these self-amplifying RNAs is that you can actually have a multicistronic presentation, so you can actually put multiple genes that are not even fused into them. So you could potentially produce like 5 different RNAs or different proteins off of a single RNA. And because of their relatively big flexibility in how much you can clone into them, that's of course a very interesting strategy if you say were to make a multiplexed RNA against multivariants, for example.
Okay. The next question is there any batch-to-batch variation? And if so, how have you controlled it?
Yeah, there is. I mean, as you kind of develop things very rapidly, your specifications kind of seem to bounce around a bit. I think as we go to larger and larger scales, it doesn't become that much of an issue. Our particle size are now coalescing within 10-15% of each other between batches. But yeah, it's certainly a concern, especially early on when you're still on the lab scale and you're still you're not as tightly controlled in your temperature parameters, for example, your cooling coils and things like that.
Yep, thank you. And then I'll just add from the microfluidic side, so long as your premix and all your formulation is kept the same, the result you'll achieve when processing through the microfluidizer should be the exact same due to our fixed geometry interaction chamber. So as long as you're able to precisely control your premixing procedure, control your temperature, the results, at least from a particle size standpoint, should be the exact same from batch to batch. The next question is the final formulation a sterile one? If so, which sterilization method is applied for clinical manufacturing?
Yeah, good question. Yeah, it has to be a sterile presentation for injection, and we do a terminal filtration. So we filter it through a 0.2 micron filter in order to get it sterile at that point. That's done in a class 100 environment and then filled into vials.
Okay. The next question is, compared to the other LNPs, does the Lion platform give appropriate RNA protections? Is the platform stability also, or does the platform stability also provide good room temperature stability?
Yeah, so as you saw, I've shown some data about room temperature stability up to 7 days. Obviously, what we're seeing now is as the RNAs get larger, the amount of room temperature stability goes down. And maybe that's not too surprising because you have to bridge presumably at some point between particles because the RNA is getting so large. So your protection isn't quite as complete. But certainly we're pretty happy with the stability we are seeing considering how labile RNAs are. Was there a second part to that question?
No, I believe you answered the entire question.
Okay.
Which brings us to the next one. So the next question is wondering if there are any side effects that you've seen from I'm assuming this means iron oxide.
No, as a matter of fact, and this again gets to this muscle localization. The side effects we've seen in the trial are much lower than what any other RNA vaccine is seeing. And we suspect that that might be due to something retaining more of those iron oxide nanoparticles at the site of injection rather than draining to the lymph node and causing, the interferon secretion and things that kind of make you feel a little terrible. Again, it's not proven. It could relate to transferrin receptors or other things that the cell puts out, but we really are still investigating that.
Okay, that's good to hear. The next one is, how difficult is it to trim the lion particles for individual formulations?
We don't really change the lion particle much at all for the individual formulations. We do optimize what we call the N to P ratio, which is the ratio of nitrogens to phosphates. So, the nitrogens on the lion particle that are mediated by the DOTAP component and impart the positive charge, they have to be coated by the RNAs, because if they're not, you get a pretty reactogenic thing, as you probably know. If you just have a lot of surface positive charge, you get cell death and you get reactogenicity. So we do tune the ratio of how we add RNA to lion, but we really don't tune the lion particle much at all.
Okay. The next question we have is, how is the encapsulation efficiency and how do you determine it? Is the non-encapsulated RNA separated out?
No, again, this relates to the optimization of the amount of lion to the RNA. So we try to get all the RNA bound so that we don't have any more free RNA. The way we measure that is we measure the amount, we add RNase and we look at the amount of RNA that's destroyed compared to the untreated. And generally that seems to be between 90 and 95% of the RNA that still is retained by the lion particles. So we think maybe it's, up to 5 to 10%, maybe that's unbound, but haven't completely proven that yet.
Okay, good to know. The next question is, which route of synthesis is used for iron oxide nanoparticles in lion platform?
It's a thermal decomposition that's done, and it's done by one of our vendors. They have a very tightly controlled proprietary process that they do. They somehow thermally degrade these particles, and then they get a very, very tight control around the average particle size. It's much better than what we see with emulsions, for example. I mean, if you look at these particles under an electron microscope, they almost all look identical. It's pretty incredible what they're doing.
That is impressive. I can handle the next question. So the next question is asking how a microfluidizer processor differs from a typical high-pressure homogenizer. And the major difference here is that our microfluidizer processors focus on constant pressure processing, whereas a typical high-pressure homogenizer focuses on constant volume processing. So again, if you can imagine our fixed geometry interaction chambers, there are no moving parts within the interaction chamber. And how this relates, if you were to look at it in a pressure profile, is the pressure profile is essentially flat, whereas for common high-pressure homogenizers, they're able to reach a similar high pressure, but because it is volume-based and there's a valve that is constantly moving, the pressure profile itself is not necessarily always flat, which means that the shear rates that your material are facing is not necessarily always consistent and the same, whereas within a microfluidizer processor, you can be guaranteed that all of your material is experiencing the exact same shear and impact forces. So I think we have time for maybe 2 or 3 more questions before we have to wrap up the webinar. So I'll see if I can pick out a couple more. An additional question was, Derek, do you have any downstream processing, for example, purification after the RNA addition?
No, the RNA addition it happens at bedside, so we don't process it after that.
Okay, that's good to know. We'll see if we have a couple more questions that we can answer here. One question was, have you done comparison studies with your own LION platform with other formulations such as Moderna's and Pfizer's mRNA vaccine?
That's a great question. You know, they don't generally send those vaccines to us for testing, but we are considering doing studies like that because it would be very nice to have some kind of head-to-head data, but we don't have the data yet.
Okay, it's good to know. And I think we'll ask maybe just one more question before we wrap up the webinar. I'll ask this one here. So the person is wondering, what is the ratio between the RNA and LION in order to form a stable LION platform, and how did you determine this ratio?
So the ratio we use, again, we depend on what we call the N/P ratio, this nitrogen to phosphate ratio, and the ratio we use right now for LION for the vaccine is an N/P of 15. So there's 15 nitrogens per phosphate in the RNA, so you can calculate, you know, how many DOTAPs are in there per nucleobase. The way we determine that is we make these formulations at various N/P ratios, and we look at transfection of the cells, as well as looking at toxicity. And you can see that there's an optimal point where the cells are transfected. And so different sizes of RNAs, as I think I mentioned before, have different N/P ratios that are optimal, but for this vaccine, it's 15.
Okay, perfect. Thank you so much. And I think we'll conclude the question and answer session there. So I would just like to again thank Dr. Derek Carter for joining us today for this webinar. There are a handful of questions that we didn't get a chance to get to. So if you do have additional questions, again, feel free to reach out to either Dr. Carter or myself. Again, our emails are shown on this slide here. Again, yeah, thank you for joining our webinar, and this will conclude the question and answer session in the webinar. Enjoy the rest of your day.